Okay, so today we wanted to talk about the physiological basis of herbicide selectivity. Basically, I'm going to talk about 3 areas. The first part, I'll be talking about the entry of herbicides into a plant and how they move within the plant. It is the entry and the movement within the plant which can result in selectivity. Then the other thing which I am going to talk about is the activity of enzymes, how enzymes can influence selectivity. That is the second thing. The third thing is about herbicide metabolism, how herbicides, when they enter the plant system, they get broken down into various products. And it is the third bit, which is a bit long. So I have displayed a cell where most of these activities are going to take place. If an herbicide enters the plant system, it may end up in the vacuole and stored there after being changed into various products. It can enter these ribosomes or rough ER or smooth ER, endoplasmic reticulum for ER. It can also enter the mitochondrion or the chloroplast, where changes may take place within the cell. That will be then the metabolism. So everything will happen in the cells. Now, when the herbicide reaches the plant, it can undergo what is known as differential penetration. Now, in young plants or young weed seedlings, the herbicide may penetrate at a high speed into the plant. It's because of the cuticle thickness which causes these differences. The cuticle thickness increases with age, which means that in the young weed seedlings, we have very thin cuticles, which allow herbicides to penetrate very fast. Most of the herbicides, like, for example, 2,4-D, it is formulated as an ester so that it is able to penetrate this cuticle readily, compared to the formulations of amine or other salt formulations. It is the entry of the herbicide inside the plant which partially accounts or explain selectivity when it comes to post-emergence herbicides which are directly applied to the plants or foliar applied herbicides. Now we have talked of the entry of herbicides into the plant. We want now to focus on their movement, or what we call differential translocation. Now with the soybean, there is a herbicide called GOR, or oxyflorphene. That herbicide moves very slowly within soybean. And it is that slow movement which can account for selectivity in soybeans. In sugarcane, we have got 2,4-D, which doesn't kill sugarcane, but it can kill beans. That selectivity is also based on the slow translocation which takes place in sugarcane, and in beans, which is susceptible, the translocation could be fast. Now, we should know that 2,4-D free acid can readily move within the phloem, and it can easily reach the site of action, which is the meristematic region. Now, if we apply the ester forms of 2,4-D, they need to be changed to acid forms, a process which we call deesterification, or we say the ester forms are deesterified. If they are applied as amine forms, they need to be deaminated to acid forms, which can readily move. It is the speed at which these forms are converted to acid forms which may determine selectivity. Well, in tolerant plant species, for example, deesterification is very slow, which means the herbicide may remain as an ester form most of the time, or an amine form most of the time, which means that it's not going to kill the plant. This structure is showing the ester form of the herbicide, which can break down into an acid and an alcohol during the deesterification process. And it is this acid which is responsible for the herbicidal activity. It can cause phytotoxicity. We can look at the example of the herbicide. This is the 2,4-D methyl ester, which can be applied to the plants post-emergence. Now they have to be deesterified, they have to undergo deesterification process. That is to break this into an acid and an alcohol. So the result is 2,4-D acid, which is the killing herbicide, and the methanol. So if this process is fast, the plant gets killed. But if it is slow, we have plants surviving. And it is this reaction which, in plants, which explains selectivity. Now, there is a process which we call compartmentalization. Now, compartmentalization means that the herbicide, when it is applied to the plant, within the plant, it is restricted to certain areas within the plant so that it won't reach the site of action and cause damage. This can explain why carrots is not killed by the herbicide called Linuron. Now, Linuron can be inactivated at the point of entry, which means it can be held there. It's not going to move in carrots, but in weeds, it can easily move and kill them. Again, we have got 2,4-D, which is a phenoxy herbicide, which can travel very slowly. It can be restricted in certain areas in grasses. This is how grasses can survive it, but broad-leaved plants get killed. We also have what is known as sequestration. Sometimes you can't separate sequestration and the compartmentalization. They all point to herbicide restriction within the plant. So in sequestration, the herbicide is kept remote from the site of action. For example, paraquat is kept remote from the site of action. It cannot reach the chloroplast, which is the site of action. This can happen in a weed known as the conizer species. And this is why some conizer species are not killed. Actually, this sort of selectivity in weeds is undesirable because it can mean herbicide resistance. So far, the physiological processes which can account for selectivity, which we have just explained, is first, differential penetration. How accessible is the plant to the herbicide? Determine whether the herbicide is going to kill that plant or not. That is the selectivity. Then if the herbicide enters the plant, how easily is it going to move within the plant so that it can reach the site of action? So depending on how that herbicide is formulated, for example, if it is 2,4-D, which may have to be converted to an acid form so that it can move very fast. It is the conversion process, the speed at which that conversion takes place will determine selectivity. Then there are restrictions. When the herbicide is within the plant, we have got the compartmentalization, like in carrots, where Linuron is restricted to certain parts of the roots. It's not going to enter the carrot system. So carrots may survive. Then we have got sequestration is also restriction of herbicides. In some, it may enter the system, but it will be restricted to some regions, like the post-emergence application of paraquat to conizer. The paraquat may not reach the chloroplast, which is the site of action. Therefore, selectivity is achieved in that way. Now, we want to talk about selectivity, which is caused at the site of action. By the way, when we say the site of action, we mean the enzymes. So it's something which has to do with the enzymes. So the enzymes themselves can determine selectivity. So we want to focus on the groups of herbicides where the enzymes are important. Basically, I will use a certain model to explain the general understanding, our general understanding of the role of enzymes in bringing about selectivity. Now, here I have displayed some diagrams showing this is the plant. If we cut the plant to the cell level, we can identify the chloroplast. So we want to find out what is actually happening at the chloroplast. For example, if we apply atrazine. If a plant is tolerant to atrazine, the enzyme will present a certain structure. For example, this D1 protein of photosystem 2, the D1 protein is actually representing the enzyme. Atrazine may not fit, or it may not be compatible with that enzyme. So it's free. It's not going to damage this crop because the enzyme is free of this atrazine. But there's a situation at the binding site of the enzyme, which we call the plastoquinone binding site. Atrazine is going to fit in at the structure of the enzyme. Once it has fitted in, then a process will take place, and that process will produce probably the mode of action of the herbicide. And that process will result in the killing of that plant. So where there's incompatibility with the enzyme, the plant may survive. So this is the general understanding of how the enzymes come into play in explaining why the herbicide may not kill or kill the plant, what we call selectivity. Now, where we have selectivity, we are talking of an enzyme which has been altered. So the enzymes may look similar, but others may look, they may be altered. And this results in selectivity. So we want to look at the herbicide groups the herbicide groups which can account for the selectivity due to these enzymes. First, we have got the triazines. Then we have got glyphosate. We have got a group called the sulfonylureas and the imidazolinones. Then we have got the Aryloxyphenoxy, carboxylic acids, and the Cyclohexenediones. All these groups can bind to specific site of action or a sequence of amino acids on the target enzyme molecule. Probably you need to understand that an enzyme is a protein, and a protein is made up of amino acids. And these amino acids form a certain structure, and it is this structure which may be compatible with herbicides, or it may not be compatible with herbicides. If it is compatible with herbicides, the mode of action of the herbicide is expressed, and usually you get a plant being killed. But if there's no compatibility, the mode of action of the herbicide is not expressed, and this results in selectivity. The plant is not going to be killed. Now, let us start with this group, Aryloxyphenoxy carboxylic acids. These herbicides under this group are known as the grass-killing herbicides. Usually we apply them like in soybean to kill grasses. They are not going to affect soybean or probably groundnuts. Now, the reason why grasses get killed, it's because of this enzyme known as acetyl coenzyme A carboxylase, or in short, AWCS, which is found in broadleaves, which could be groundnuts or soybeans, and grasses, which could be weeds. Now, in grasses, this enzyme combines with the herbicides. After the combination, you get a disruption of a process known as the lipid synthesis, and this will cause the grasses to be killed. But this cannot happen in a broadleaf plant, and therefore the broadleaf plant is going to survive. So basically, we have got, let's say, groundnuts, soybean, and the grasses sharing the same enzyme, but in broadleaves, that is the soybean and the groundnuts, this enzyme is not going to combine with the herbicide. But in grasses, there's going to be successful combination, which results in the killing of the grasses. Here I've got herbicide examples. We have got fused lead, which is normally applied in soybeans or groundnuts to kill grasses. The other name for fused lead is fluoxyphop-butyl. And we've got another herbicide called Pilot Super. The chemical name is quizalofop-ethyl. We also have another herbicide called the Holon, which is dichlorphop-methyl. These herbicides are grass killers in broadleaf crops. We know why they kill grasses. It's because they can combine with enzymes in the grasses to express their mode of action, which cannot happen in soybean and groundnuts, That's why they fail to kill those crops. We also have some commonly used herbicides like NAP, cetoxidin, and select clethodim. These fall under the group which is known as the cyclohexenedione herbicides. They are also grass killers, and the enzyme is the same, which is acetyl coenzyme A carboxylase. In grasses, they can successfully combine with the acetyl coenzyme A carboxylase. But this cannot happen in broad-leaved crops, which results in selectivity. So they can also be used to kill grasses in broad-leaved crops, soybean and groundnuts. Now, coming to triazines, we said that things like atrazine may combine with the enzymes and produce phytotoxicity. But in some altered enzymes, there's going to be failure to combine, especially at the quinone binding site. This is why some resistant weeds are not killed, because atrazine will fail to combine at the quinone binding site. Then we move on to sulfonylurea and imidazolinone herbicides. Again, we have got an enzyme which is known as acetolactate synthase, or in short, ALS. Now, a change in amino acid sequence on this enzyme can prevent the binding of sulfonylurea and imidazolinone herbicides. And of course, this can result in selectivity. The other name for the acetolactate synthase enzyme is acetohydroxy acid synthase, AHAS. Now, these are the sulfonylurea herbicides: chlorsulfuron, primisulfuron. Nicosulfuron is the accent, which is used to kill chafer grass in maize. As far as the imidazolinones are concerned, we have got imazapa, imazamox or lynx. Imazamox or lynx is commonly used in groundnuts in this country, sold by other agrochemical companies. Imazatapa is besut, is also commonly used to kill grasses in soybeans. Accent or nicosulfuron, it's the herbicide which is used to kill chafer grass in maize. Now, here we have got a glyphosate. Glyphosate can inhibit an enzyme known as 5-enolpyruvyl shikimate 3-phosphate synthase, EPSPS. In short, the altered EPSPS, which is done artificially in GMOs, enable us to use glyphosate to kill weeds in maize and the soybeans. So there is genetic engineering which has been done on some crops to create GMOs. What they have just done is to create EPSPS, which is not compatible with the glyphosate. That's how that selectivity is achieved. So I wanted to move on to the third part. Remember, so far I have explained selectivity first by talking about how herbicides enter the plant system and how they move within that plant system, the speed at which these herbicides move, and how they enter the plant can account for selectivity. Then the second thing is the activity of the enzymes, how they differ in grasses and in broad-leaved crops, which can also account for herbicide selectivity. So we have just talked about the enzymes. Now we're going to move in a different area. We want to talk about herbicide metabolism, how herbicide metabolism can result in selectivity. So what is herbicide metabolism? Now suppose the herbicide is successfully entered the plant system. Inside the plant, that herbicide is going to be destroyed or degraded. Probably in tolerant plants, the herbicides are degraded very fast. Whereas in susceptible plants, this degradation of herbicides is slow. And it is the degradation of the herbicide which will influence its concentration at the site of action. What we are saying is that if you have degradation being slow, it means you've got low amounts of herbicide at the site of action, and the plant can easily survive. Now, on metabolism, or herbicide metabolism, there are 4 areas we are going to talk about, which can account for pesticide degradation, or what we may call detoxification. It is the detoxification which imparts selectivity, or which causes selectivity. First, we have got oxidation here. Then second, we have got hydrolysis. Then third, we have got reduction. And the last thing is conjugation. You are going to realize that these processes are classified in phases. Phase 1, phase 2, and phase 3. Actually, oxidation, hydrolysis, and reduction are part of phase 1, which is quite a long process, and the conjugation can be divided into phase 3, phase 2, and phase 3. So I'll explain what is oxidation. Then I'll move on to hydrolysis, then reduction, and at last I'll talk about conjugation. So when a herbicide enters, or when the herbicide enters the plant system, it is subjected to 3 phases, depending on the molecular structure. In phase 1 reactions, there's going to be oxidation, reduction, and hydrolysis. In phase 2 reactions, there will be conjugation reactions. Phase 3, there will be there will also be conjugation, but the conjugates are moved to the vacuoles, or they become part of the cell walls. So in short, this is what I'm going to talk about. So let us talk about phase 1. The herbicide has entered the plant system. We want to see the function and the roles of phase 1. Now, phase 1 reactions are catalyzed by what is known as the cytochrome P450 monooxygenase, which occurs in the endoplasmic reticulum. Remember, I showed you a cell in the beginning where you can find this smooth ER or rough ER. ER representing the endoplasmic reticulum. Sometimes the cytochrome P450 monooxygenase, there are a lot of them. They are called mixed function oxidases. In general, these enzymes protect organisms from toxins. They catalyze oxidation reactions. What happens is that oxygen is incorporated into the herbicide molecule. The oxygen atom is converted into water by an electron donor, which is NADPH. So what we are saying is that in the phase 1 reactions, we need cytochrome P450 monooxygenase. This is an enzyme which is going to catalyze most of the reactions which take place in phase 1. I just wanted to give you an example of a reaction which is catalyzed by cytochrome P450. Now, RH in this equation represents our herbicide. And we have got NADPH plus hydrogen ions plus oxygen. These are requirements to change the RH herbicide. After the reaction, we get NADPH plus water, and ROH. ROH is the altered herbicide, or it is the herbicide metabolite. So RH represents the herbicide, ROH represents the herbicide metabolite or changed herbicide. By the way, it is not only herbicides which are altered when they enter a living system. We have all sorts of substances which can enter a living system. And all these substances may be regarded as xenobiotics or foreign substances. But of course, our focus is on herbicides. What can happen when they enter a plant system? Now, in phase 1, with the aid of cytochrome P450, there's going to be formation of functional groups. It depends on the nature of the herbicide. It can be converted to OH, NH2, SH, COOH, CN, COCH3, CH3, or SCH3. All these in red are known as functional groups, and these can be added to herbicide molecules. Once the functional groups are added to herbicide molecules, there is going to be reduction in phytotoxicity. No wonder why in some plants, the herbicides may not kill these plants, that is in crops. So in phase 1, let us remember that functional groups are formed. Now, basically what happens in phase 1 is the oxidation reactions. So we will explain what is oxidation. What is it? Oxidation is comprised of 5 processes. Number 1, we've got what is known as hydroxidation. Number 2, we've got N-dealkylation. Number 3, we've got O-dealkylation. Number 4, we have got oxidative deamination. And number 5, we have got Self-Oxidation or thioether oxidation. So 5 things under oxidation. And these processes are the ones which can produce functional groups or add functional groups to the herbicides so that they lose phytotoxicity. So what we are going to do is to unpack oxidation. We will start with the hydroxidation. Now, hydroxidation, it means adding the OH group to the herbicide molecule. For example, we can have an aromatic group, which is a benzene ring carrying a chlorine atom. This atom can be substituted by an OH so that this active herbicide is no longer active when we have OH. If it is an aliphatic group, where we can represent it by R attached to Cl, we can convert it to ROH. This reaction of adding OH to these groups is what is known as hydroxidation, and it is catalyzed by cytochrome P450. Here I have got a Basagran hydroxylation. Please don't worry or don't be scared by these chemical structures. I just want to show you what can happen to this big structure of Basagran. What has happened is that OH group has been added on this benzene ring in the presence of phytochrome P450. It's a hydroxylase because it causes hydroxylation. Now, once you have added OH, Basagran will lose its phytotoxicity. This could be happening in soybean because when we apply Basagran to kill weeds in soybean, most of the broad leaves are killed, but in soybean, Basagran does not kill soybean. So the selectivity in soybeans is happening because of this addition of OH to the benzene ring, which is happening very fast, causing Basagran to be less phytotoxic. If you don't want to, if you are worried about the chemical structures, we can simply represent Basagran by this circle. We write Basagran and we say hydroxidation. We add OH to this circle. Then if that circle is carrying OH, it means that Basagran is no longer phytotoxic. After hydroxylation, the other reaction is N-dealkylation. For any dealkylation to take place, we need this structure. R is representing the complicated structure. We just want to focus on this NHCH3. This is what is important. So after any dealkylation, we are now having this NH2, which is an amine. I'm going to show you where this can take place. We've got atrazine, and the atrazine has got these structures with N. So this structure can be converted to NH2 here. Again, this other part can further be converted to NH2 in the presence of P450, cytochrome P450, the enzyme. We can simplify the structure by writing this to represent the atrazine so that we get this NH2 attached. The other process is known as the O-dealkylation. We have this sort of structure, R representing the big part of the molecule, and OCH3 is the important part. After O-dealkylation or removal of the methyl group, we are now having R OH. I'm going to use 2,4-D acid to demonstrate O-dealkylation. Where you see this arrow, red arrow, this is where this part is going to break and form an OH. So this is O dealkylation, removal of this bit. Self-oxidation or thioether oxidation, the fifth process under oxidation. For this to take place, we should recognize this bit where we've got R representing a big part of the molecule, but it's the S attached to CH3, which we should recognize. And oxygen is added to sulfur. Then that process is self-oxidation. We have got prometrine with this structure. Now, if self-oxidation takes place, we are just simply going to add oxygen at the sulfur molecule. And this detoxifies prometrine. Of course, you can see that we also have these structures which can undergo N-dealkylation. So the same molecule can undergo self-oxidation or thioether oxidation followed by N-dealkylation. Here we have got EPTC. Don't worry about the complicated structure. Just focus where you see S. Where you see S, we are going to add oxygen. And this causes EPTC to be less phytotoxic. Or we can represent it here down there. Where we have S, I have circled the important structure. If we add oxygen, that is self-oxidation. And this will cause this herbicide to be less phytotoxic. EPTC is usually incorporated in the soil, and it controls sedges or nut grass in maize. There's also oxidative deamination. Now, for oxidative deamination, I think this is the last process. You need to have two nitrogens in a chain attached to this R, and it can break, leaving RNH2. This can happen on a metribuzine molecule. We've got this metribuzine. Don't worry about this big structure. But where you have double N, we can split this. Then we are remaining with hydrogens, which are attached here. This can reduce the phytotoxicity of metribuzine. But of course, we do have also something interesting on metribuzine, which is SCH3. There's also a possibility of undergoing self-oxidation or thioether oxidation on the same molecule. Now we have finished with the oxidation with the 5 processes. Now we want to talk of reduction, which is just one process. With reduction, there should be NO2 attached to R, and then this should be converted to NH2. Of course, under cytochrome P450. We've got the herbicide called Trifluorine. This is a grass killer. Normally, it is sprayed on the soil surface and then incorporated. Usually, it controls grasses in cotton and the soybeans. So this is its structure. it has this NO2 on it. These are converted through nitro reduction or a reduction process to NH2. Or we can simplify it down here, where we have R representing the complicated structure, and NO2, what is important, is converted to NH2. But if you look at the structure of trifluorine, there are possibilities that we can break it here by N-dealucylation, which can take place twice, on the left, on the right, and on the left. And also there's this bit here where we can get OH being attached. So reduction is one process. Then we've got hydrolysis, the addition of water, which can lead to breakdown of herbicides. This can happen if we have got an ester or a nitrile or amide groups. This bit shows what an ester looks like. It can break down to an acid and an alcohol. For example, we've got 2,4-D methyl ester. We talked about this in the first part. This can be de-esterified, or it can undergo de-esterification into 2,4-D acid and alcohol. The speed at which this is happening can cause phytotoxicity. So this is ester hydrolysis. There's also what is known as amide hydrolysis. The amide structure is this, where you've got R, NH being attached to carbon, then we have got oxygen here, and then another R. So this is part, if you can identify it on a herbicide molecule, you can predict that it can undergo amide hydrolysis. Here we've got the NH2 attached to R. So the amide hydrolysis, we've got propanil. Don't worry about the complicated structures, but just focus on the amide part, which can break down into an amine and this propionic acid or just acid. The propanil is a herbicide which is used to control weeds in rice. So in a rice plant, it can easily break down through amide hydrolysis. Another hydrolysis can take place in when a herbicide has got what is known as the cyano group. The cyano group is a triple bond between carbon and nitrogen. If we have got this structure in any molecule, it can undergo hydrolysis. So again, it is this structure, and we call this nitrile hydrolysis because of nitrogen here. First, it forms a carbamate. This is a carbamate. It can further break down into an acid and ammonia. I will then show you the herbicides which can undergo nitrile hydrolysis or just hydrolysis. We've got Bladex. Bladex is the herbicide which is used to control weeds. in maize. If you look at the structure of Bladex, this complicated molecule, it has got this bit here, which is called the cyano group. Firstly, it will be converted to an amide. Then later, it's converted to carboxylic acid. So the process is hydrolysis. Addition of water to the molecule, then it can cause these changes. Bromoxynil or Bactrim is the herbicide which is used to control weeds in wheat. It also has a structure with the cyano group. If you add water, you convert it to carboxylic acid. That's how it can lose its phytotoxicity. So remember, Bactrim is used to control weeds in wheat. So under phase 1, all these reactions are promoted or catalyzed by cytochrome P450. Now we move on to phase 2 reactions. It has been a long journey in phase 1. Now in phase 2, we are talking of conjugation reactions. One of the most important detoxifying molecule which can result in selectivity is known as glutathione. That is an important molecule. What glutathione does is going to utilize some of the products from phase 1. So after all the reactions in phase 1, at the stage 2, glutathione will now utilize some of them. And there's also an enzyme which can catalyze these reactions. The important enzyme is the GST. The long name is glutathione S transferase. So you've got glutathione S transferases because there are many of them, which work in conjunction with the glutathione. They will then use all the products or some of the products from phase 1. What is interesting about glutathione is that it has a reacting component known as the SH. That is the one which can attach itself to the products from phase 1. So what is glutathione? GSH, if you want to call it in short. It's basically a protein, a tripeptide with 3 amino acids, namely glutamate, which is joined to cysteine, and cysteine joined to glycine. So we can represent it in short like glu-CYS-gly. So this bottom part here can represent the short structure for glutathione. I've got an example of glutathione conjugation reaction. This is the altered herbicide from phase 1. Maybe what has happened is hydroxidation. under the influence of cytochrome P450 in order to produce this herbicide with an OH. Now, when it moves to the phase 2, glutathione will take over and react with this component. And the reacting component is the SH, which is attached to cysteine, the middle amino acid. Remember, glutathione has got 3 amino acids. Glutamate joined to cysteine, cysteine joined to glycine. And it is the cysteine which carries the reactive component, SH. So what is going to happen is that this OH of this herbicide is going to link to the SH to form this structure. So you now have the herbicide part which is joined to the S part. What is the product? We call the product glutathione conjugate. So your herbicide is now a glutathione conjugate. Probably the glutathione conjugate will now be moved to the vacuole for storage. And that will be the end of this herbicide. So glutathione conjugates are now transferred to the vacuoles for storage. And that's the end of the herbicide. Here I'm going to give you an example of atrazine because in maize, the selectivity of atrazine is basically based on the glutathione activity. In fact, glutathione will not wait for atrazine to be converted to an OH in phase 1. This process can just start in phase 2, where you have glutathione with this reacting component substituting the Cl, and then you have this structure where you have glutathione attached via the sulfur bond to what was then atrazine. And this is facilitated by the enzyme glutathione S transferase. So once this has happened in maize, and in maize leaves, this process is taking place at a very fast rate, conjugation process of the atrazine being done by glutathione. That's why atrazine is not doesn't kill maize. We have talked about glutathione, how it can cause selectivity. We know that sugar in an organism or in plants, it is supposed to provide energy via this respiration. But another function is that sugar molecules can protect the plants from herbicides effects. So there are certain herbicides with the following functional groups. If in phase 1 we have got a herbicide carrying NH2, which is an amine or an N-aramine or just an amine. The second, we can have a herbicide carrying the carboxylic acid or COOH, or we can have phenols or OH. Such products of cytochrome P450 activity in phase 1 will be further acted upon by sugar molecules to further change them into other things. So we will see how sugar molecules can further react with these products. So what basically happens, we are going to form sugar conjugates. Uridine diphosphoglucose, or UDPG, provides the sugar, or it is the sugar donor. And during that process, we have an enzyme which will catalyze the process. It's known as the UDP glucosyl transferase. So UDP glucose, or UDPG, or UDP, or uridine diphosphoglucose, let us just call it UDP glucose, or simply UDPG. That is the sugar donor. Now I want to talk about the reactions which can take place. The reactions are basically known as the glucosylations. First, we will talk about what is known as O-glucosylation. Now, you have the herbicide carrying the OH. In O-glucosylation, you have UDP glucose, which is the donor of glucose. It will provide the sugar, which is going to react with this herbicide carrying an OH. Once it has donated the glucose, UDP is formed. Now, glucose is going to be attached to the herbicide via the oxygen component. And the whole process is facilitated by an enzyme known as glucosyl transferase. So we now have this glucose or herbicide, which is no longer a herbicide. Here, I just want to show you what can happen to Basagran in soybean. This product carrying an OH attached to the Basagran molecule. Remember, the active Basagran doesn't have an OH, but through the activity of the cytochrome P450 in phase 1, it's now carrying an OH. What is going to happen is that UDPG will donate glucose to this entity of Basagran. Then they are going to form this structure, which is quite complicated, which is glucose or Basagran. Glucose or Basagran. And it's facilitated by UDP glycosyl transferase. So in soybean, this is basically what is happening. You have cytochrome P450 adding OH to the Basagran molecules, and UDPG taking over by providing the glucose to this derivative to form glucose or Basagran, which is now no longer phytotoxic. We move on to another herbicide derivative from phase 1, a product also of cytochrome P450. UDP glucose will donate the glucose and form UDP, and the structure which is formed is glucose N herbicide. And you need glucosyl transferase to catalyze this reaction. So in the second part, we now have glucose N herbicide. In the first part, we had glucose O herbicide. Then we have got a situation where we have this product, herbicide, the COOH, a product of cytochrome P450 in phase 1. UDP glucose adds glucose here, and it's now UDP. Now glucose is now added to this part, and it has this carbon which is attached to oxygen, and this is done via glycosyl transferase. So this is no longer an active herbicide. It's now fixed. We can use the example of 2,4-D, which is an acid. So it can actually be cleared by glucose. UDPG donates glucose, and this glucose gets joined to this herbicide. So you now have a 2,4-D or glucose, or the glucose ester of 2,4-D, which is no longer toxic. The other important and interesting thing is we have amino acid conjugations. The most important amino acid is known as aspartate. Aspartate will require herbicides carrying a COOH or a carboxyl group. Also remember that herbicide carrying this acid are products of phase 1 under the influence of cytochrome P450. Then in phase 2, they are undergoing further changes via aspartate. So aspartate can combine with these to form these structures. Herbicide, which is combined to the aspartate via the carbon with a double bond to oxygen and this NH. So this is the actual 2,4-D, and this is the actual aspartate, and this is how they are joined. But there's no need to remember these structures, only to know that 2,4-D is joined to aspartate, and it can lose its herbicidal activity. Remember, we're talking of how the plants can get rid of these herbicide molecules within the systems. Then we move to the last phase, which is phase 3 reactions. Remember, plants do not excrete herbicide metabolites like what animals do. After ingesting a pesticide, the pesticide can be excreted via urine or sweat. But with the plants, these metabolites are stored in vacuoles. So in phase 3, we need further conjugation reactions utilizing products from phase 2. Then these products are then moved into vacuoles for storage, or they can be incorporated into the cell wall region. Herbicide glucosides are further processed by addition of malonic acid residue to sugar conjugate. Let's see what happens. Already we've got a glucose O herbicide, which is a product of phase 2. We can add malonate or malonyl coenzyme A to this bit. Then we get malonyl joined to glucose, joined to herbicide, and the coenzyme A is released. Now we have this product, which either it can be incorporated into the cell wall, or it can go to the vacuoles. So in phase 3, we are further building up complicated molecules. For example, glucose, where we add malonyl to get that structure. So we add either malonyl coenzyme A or malonate to glucose O herbicide to produce malonyl glucose herbicide, which is then become part of the cell wall, or it can go into the vacuoles. Again, there's something called the joint biocide formation, where you add further glucose conjugations. You add glucose to glucose herbicide, then you get the result of glucose herbicide. That structure is no longer toxic. So that will fix the herbicides. It is the speed at which these herbicides are being altered from phase 1, 2, and 3, which will account for selectivity in plants. If these processes are slow, some plants will get killed. But if these processes are fast metabolism, the plants will survive. Well, let me give you a summary of where we came from and where we ended. In trying to understand the physiological basis of herbicide selectivity, we talked of entry of the herbicide into the plant, the cuticle effects. If you have got a young plant, there's fast penetration compared to a mature plant. Then there's herbicide movement within the plant. Can it reach the site of action? Depending on how it is formulated. Remember, some herbicides, you need to convert them to the acid form. It is the rate at which they are being converted to an acid form which will determine selectivity. There's also compartmentalization, restriction of herbicides. Same applies to sequestration. This is also a restriction of herbicide movement, which can result in selectivity. Then we talked about the role of the enzyme. And we said enzyme structures can bring about selectivity for a certain group of herbicides. For example, triazines, the plastoquinone binding site, glyphosate, the EPSP synthase, then the Aryloxyphenoxy carboxylic acids, and the cyclohexenediones. Specifically, the enzyme which is important is acetyl coenzyme A carboxylase, or ACCS, which can determine selectivity in broad-leaved crops and grasses. Then we also have sulfonylurea and imidazolinones, where we have acetolactate synthase, or ALS, as the enzyme being important. In all these herbicide groups, it's the enzymes which are causing selectivity. In some cases, they actually account for herbicide resistance in grasses. That is the sort of selectivity which the farmer may not want. Now, we also discussed the selectivity which is brought about by herbicide metabolism, which is classified in 3 phases. With phase 1, we talked about the activity of cytochrome P450, how it enables the formation of functional groups, which are OH, COOH, NH2, SCH3, SH, and OCH3. Remember, it is these functional groups which are required in phase 2. Basically, phase 1 is the oxidation process. That is number 1. Number 2, it is the reduction process. Number 3, it is the hydrolysis. But oxidation has got 5 processes, namely hydroxidation, and dearyloxyphenoxy, or dearyloxyphenoxy, Oxidative Deamination, and Self-Oxidation or thioether oxidation. Reduction is just one process where you are reducing NO2 to an amine. Hydrolysis basically may have 3 processes where you are breaking down esters, amides, and nitriles using water. Then in phase 2, we talked of conjugation reactions. Basically, there is glutathione, which is composed of 3 amino acids, glutathione, which is joined to cysteine, cysteine, which is joined to glycine. And there's an important enzyme which facilitates the reactions, which is known as glutathione S transferase. We also talked about the role of glucose, which is donated by UDPG, or glucose comes from UDPG, and the enzyme which facilitates the reactions is known as UDP glucosyl transferase. Basically, glucose will act upon the derivatives from phase 1 herbicides with a an OH, NH2, and a COOH. Then we also have an amino acid, which is aspartate, which can act on herbicides carrying a COOH. Remember, these are now altered herbicides. And finally, in phase 3, where we have malonate or malonyl coenzyme A, malonyl is added to glucose herbicide, resulting in malonyl glucose herbicide. Or you can have a further addition of glucose, then you end up having a glucose-glucose herbicide, which is known as agent biocide. So these products can either be stored in the vacuole or they can become part and parcel of the cell wall. And this is how the herbicides are destroyed in plants, resulting in selectivity. Well, it has been quite a journey in trying to account for herbicide selectivity. So that is it. That is the end. I will stop sharing. Hello, are you still there?
Hello, Doc. I'm still around.
Well, it has been quite a long. I wanted to break it down, but well, I thought let me just go on.
Yeah, it wasn't too long, but only the factor of terminology and a little bit of processes.
Yeah, at some stage we may have to discuss certain. Well, which areas did you find or you have forgotten?
I think some of the things they need revisit completely, but they are not that tough.
Yeah, but
The concept
basically
of
what you
gas.
have to know is that there are three things on a physiological basis of herbicide selectivity. The first part is we are talking of how can the herbicide enter the plant.
Mm-hmm.
The first thing is the barrier, which is the
So,
cuticle. If the herbicide succeeds in entering that cuticle, then perhaps it is going to cause some damage. But probably selectivity begins at the cuticle level. Because some herbicides may not enter that plant. Now, the second bit is, should that herbicide enter the plant, how is it going to travel within the plant? In what form? Now, some herbicides like 2,4-D, although they are applied as esters in order to break that cuticle, once they are inside, esters cannot move very fast. So they will need to be converted to the acid form. So again, it is the speed at which that conversion takes place to facilitate transport so that they reach the site of action. Again, there are other things within the plant. You know, we talked of compartmentalization, restricted movement, sequestration, which can also restrict movement. So all those things can account for selectivity. Remember, we are dealing with the various chemicals, and you can use examples here and there to explain these movements. Then apart from that, the second issue is the activity of the enzymes. You know, there are some herbicides which can kill grasses selectively in broad-leaved weeds. Selectivity here is governed by the type of enzymes which exist within grasses and the broad-leaved plants. And we went through herbicide families which are affected or which are influenced by these enzymes. Then the long journey on selectivity is on herbicide breakdown or metabolism, which is divided also in three parts. Phase 1, which is driven by cytochrome P450. Then phase 2, where you need now things like glutathione, glucose, and the amino acid aspartate. Then finally, in phase 3, we only talk of malonyl coenzyme A or malonate, which further changes the glucose or herbicide, where you now have malonyl glucose or herbicide. Or you can have further additions of glucose, where you have glucose, or herbicide, something like that. And all of these things, they are either stored in the vacuole or they become part and parcel of the cell wall. So remember that all these activities are happening simultaneously at fast rates in some crops or weeds, where these things are happening very fast, plants may survive. And where you have slow processes, you know, there are a lot of things which are happening. Either the plants are going to die because the herbicide will be very active. So this is what is accounting for selectivity, in short.
Okay, Doc.
When it comes to the structure of the herbicides, there's no need really to remember the molecules. What you need to spot is the reactive parts, which I said were functional molecules, which are derived in phase 1, like NH2, COOH. Those are the things which can participate in a reaction. So if you are presented with a molecule, if you are asked, how is it likely to break down? If there is a sulfur, do you know what is going to happen to it? If you spot a sulfur element in that molecule.
Adding hydroxyl.
We talked of self-oxidation, remember?
Yes.
Yes. We can add oxygen to the sulfur. Once we have done that, we have already changed the molecule. So there are certain bits which you need to spot on the molecule to understand how it can actually break down.
Okay.
Rather than memorizing, you know,
the whole thing.
Like metribuzin. There's no need to memorize metribuzin. But to understand that there is sulfur somewhere on metribuzin, which can cause self-oxidation, or there's a bond between nitrogen and another nitrogen, which will cause another process. Right. Any questions?
So, Doc, it
Yes.
means all these processes that we're discussing can also lead to herbicide resistance in some plants or some other grasses or some other
Yes.
weed species.
Yes. It can. What we call selectivity in crops could actually turn out to be resistance in weeds.
Okay.
And this may not become desirable for us.
Okay. So, in crops, sometimes it happens naturally, and sometimes it has to be initiated by us humans, just like maybe doing crop breeding and the genetic modification of
Yes,
the
yes.
crop genes.
Like the GMOs, it's a deliberate way. We have glyphosate, we have this EPSPS, which was genetically modified to make maize and soybean resist glyphosate, or to cause selectivity in soybean and maize.
Okay.
Yeah. But sometimes it will turn around to be undesirable later.
Okay.
Mm. Any other issues?
Maybe from Grace and Wimpai.
None from me, Doc.
Okay, so, but when I ask you questions, I don't ask you to memorize those structures. I go for just concepts.
Okay.
Yeah. So I have already sent you some notes, isn't it? For this one.
No, today you didn't send anything.
Oh, so I'm going to send the notes, and later on I will send you the recorded lecture so that you use those in conjunction.
Okay.
So tomorrow we are not going to meet, isn't it? Because it's too much now.
Yeah, tomorrow tine humwe urongwa huripo.
Yeah, at least now with this, I think we will meet on Monday.
Okay.
Anyway, it will also give you time to think it over before we move to the next.
Okay, Doc.
Okay, thank you very much.
Okay, goodbye.
Goodbye.